Hello everyone, Gayatri Prem Kumar here, and today we are going to discuss about one of the slide preparation, that is study of pollen germination, and this experiment carries 5 marks. Now in this video we are going to see what is the introduction, what is the knowledge about the pollen that we have, and then what is the aim of this experiment, materials required, procedure employed, and what you observe finally, precautions to be followed during this experiment. So this experiment carries 5 marks. We have two slide preparation. One is the pollen tube germination. The other one is the study of mitosis in the onion root tip. I have given the link in the description for the study of onion mitosis and onion root tip. You can have a look at that video. And this video we're going to discuss about all these key points and how to get full 5 mark for this experiment. Shall we get into video, children? And now we get this video. Now first thing is introduction. So when we talk about the introduction, first what is pollen? Where is this pollen present? We all know that a flower contains a male part and a female part, or the other way. A flower may contain male or female. Now coming to the male part of the flower is called as the stamen, and this stamen is divided into two parts, the anther and the filament. The slender stalk-like region is called as the filament, and the bilobe structure is called as the anther, and that anther contains a sac-like region called as pollen sac, and inside that you have the pollen grains. Got it? Now what we are going to do, in that pollen grain contain the male gamete. So this male gamete will switch on the stigma of the female part of the flower, and this male gamete will reach the ovary. The female part of the flower is called as ovary and then ovule, and get fertilized. Now we are going to see how this pollen grain form a tube-like structure, and that tube-like structure, through the tube-like structure only the male gamete will travel to reach the female gamete. Now we are bringing an experimental setup in such a way that this pollen tube formation takes place. So what do we require? So this is the basic introduction about what is the pollen, and what we are going to do is we are going to create a nutrient solution, and this nutrient solution we dust the pollen grains from the flower. We just dust the pollen grains from the flower. It could be any flower, it could be any seasonal flowers, it could be vinca rosa or hibiscus. We just take the seasonal flowers which are freshly plucked, because as time passes, what happens, the pollens will not be viable. So only if it is viable, and when you provide the nutrient medium, the germination process will take place. So you pluck the flower, seasonal flowers, and immediately fresh flowers, you immediately place it in the nutrient solution and follow the procedure. So this is about the introduction, and what is the aim of the experiment? To study the pollen germination on a slide, and this slide is observed under the microscope because you cannot see by the naked eye. Then what are the requirements that we require for this experiment? Obviously, we require fresh seasonal flowers, and then the slide where we place a nutrient solution, and then we dust the pollen, tease it nicely with a teasing needle, place a cover slip on top of that, and observe it under the microscope. So we require a microscope, and then the sucrose, boric acid, magnesium sulfate, potassium nitrate, all these are the components which make up the nutrient solution. So these are the basic requirements, and what you observe under the microscope. So what you observe under the microscope is, this is the pollen, structure of the pollen, and anyhow you have the detailed description about the pollen which you have studied in the sexual reproduction flowering plants, your theory part. And the outermost irregular layer is called as the exine, and the innermost continuous layer is called as the intine, and these openings are called as the germ pore. And when the nutrient solution is placed, this pollens will absorb the nutrient, and thereby they form the pollen tube. The pollen tube formation takes place. Now, what is the purpose of preparing the nutrient solution? So, nutrient solution is prepared by adding sucrose, boric acid, magnesium sulfate, and potassium nitrate. And what is the role of these components in the nutrient solution is, it promotes the pollen tube formation. It enhances the pollen tube formation. So, thereby, when you place it in the slide, the nutrient solution, the pollen start to absorb, they take in the nutrient solution and induces or enlarges the pollen tube formation. The triggering the pollen tube formation takes place because of these components present in the nutrient solution. Now next we will see what is the procedure employed, how you have to observe the pollen tube germination under the microscope. Now, what is the procedure employed? So, already I have told you, we prepare a nutrient solution by using boric acid, sucrose, magnesium sulfate and potassium nitrate. So, from this nutrient solution, we place a drop of this nutrient solution on a clean glass slide, and we take any of the seasonal flowers that is available. As I told you, Vinca rosa is the flower which gives a very good result for pollen tube germination, or any other seasonal flowers if you want, you can take whether hibiscus or any other flower. And then you have to dust the pollen, and that pollen has to be teased, spread nicely from the anther in that nutrient solution on a clean glass slide. Then you have to place a thin transparent cover slip on top of that, and then you have to leave it for minimum 5 minutes. So immediately 5 minutes you cannot see, but you have to leave it for some time. From 30 minutes to 1 hour, you can see the pollen tube that germinate. And again, the pollen tube formation also vary from one flower pollen to the other flower pollen, because the pollen tube formation is not an easy process. As you can see, the outer layer is there, exine, and inner layer is intine. So through this germ pore opening only, this wall disintegrate, and thereby the pollen tube formation takes place slowly. So the pollen tube formation will vary from one type of flower to the other type of flower. Generally, 5 minutes you have to wait and start observing under the microscope how is the structure of the pollen and how the pollen tube slowly start forming. Then you leave it for some time, and then after 30 minutes again you keep the slide and observe for the pollen tube formation. Up to 1 hour you can wait and observe it. Now this is what you observe under the microscope, and the pollen tube formation from one of the germ pore. And how to handle the microscope, that a link I'll be giving you in the description, because though you handle the microscope in your class, many of you have doubt how to handle the microscope, through how to keep the slide. Sometimes no, you have that when you rotate the eyepiece, you can see the image also rotating, so you will not know which is the field to capture it. And how sometimes when you're focusing, what happens, you break the slide because you bring very close of the lens. So the parts of the microscope, how to handle the microscope, so that details I'll be giving you in another video, and the link will be there in the description. You can have a look at how to handle the microscope. And the next thing is about, this is what you observe. You can call the examiner, either the internal examiner or external examiner. You can call them and show the pollen tube that you have formed, that is formed in the slide. You can show it to the examiner. And since it is a slide preparation, obviously you will be drawing the diagrams in your writing part of your practical examination. And what are the precautions that has to be followed? So as usually I have told you, you have to observe under the microscope and 10x and 45x, and how to handle the microscope, that details I'll give you in the description link. Then what are the precautions that has to be followed? I told you already, the flower should be freshly plucked. Why the flower should be freshly plucked? As the time goes, as the time extends, what will happen? The viability of the pollen will be less. So thereby the freshly plucked flower should be used for this experiment. And the clean glass slide. Why the clean glass slide? Sometimes the dust particle present may interfere with your observation under the microscope. So keeping these things in your mind, you have to take the a clean glass slide or any other apparatus that you're using for the experiment. Hope you have some information about this experiment, which is one of the slide preparation for your practical examination carrying 5 marks. And here you have an introduction about what is pollen, because viva questions will be asked in such a way, like what is pollen, what is the structure of pollen, and what are the components present in the nutrient solution, what is the role of each component present in the nutrient solution, and what are the different flowers we use for the pollen tube germination, what is pollen tube, and what does the pollen contain? It contains the male gamete. And what is the purpose of this tube formation? It has to go to the female gamete, and that is the purpose of the pollen, because the pollen comes and sits in the stigma of the female flower. So this pollen will form a tube-like structure and goes to the ovary and then reaches the ovule. So these information you should have an idea. And moreover, what is the procedure employed and what do you observe under the microscope? Parts of a microscope, what is the principle behind the microscope? And then the observation part, the pollen tube forms from one of the germ pore. And following that, the precautions that has to be followed. So hope you had some information about this video, and thank you for watching.